So, good morning, everyone. I hope the coffee is working by now. My name is Spyros Darmanis. I work at CZ Biohub, and today I'm going to tell you a little bit about our work on large-scale single-cell transcriptomics, and I'm going to specifically talk about Tabula Muris, which everybody pronounces in a different way, like my name. So, I know that Steve already talked about this. I just want to give a little bit of context as to, what I am and what I do. As Steve mentioned, one of the pillars of Biohub is the internal research, and that is basically outside the platforms divided into two large initiatives, the infectious disease and the cell atlas. I find myself working on the cell atlas side together with Loic Royer and Manuel Leonetti, who are going to be presenting some of their work later today or tomorrow. Now I'm just going to talk to you about the cell identity part of what we do. So over at Biohub, one of our main focus is basically trying to understand the underlying mechanisms of disease and then hopefully develop new technologies and strategies that will lead into some sort of actionable diagnostic or therapy or treatment or management in the future. Taking a small step back, in my view at least, understanding disease implies that we first understand the differences from the normal state, and then we try to infer what went wrong, and then try to do something with that knowledge in order to intervene, treat, and manage. Now, the problem is that in many cases, a molecular understanding of the normal state might be lacking. And by molecular understanding, I mean, what cells are there, how do they talk to each other, all the beautiful stuff that Ido just talked about. In many systems, we're just beginning to discover these. So the question that we have over at Biohub, or that we had some time ago is, all right, so where do we start? What is a good first thing to do? And to improve our understanding of the normal state and then move on from there. And we thought, among other things, that building, annotating, and analyzing data within a transcriptional reference of all major mouse tissues with single-cell resolution is probably a good place to start. So we did that, and today I'm going to talk to you about this. So we call it the Tabula Muris. This is fancy Latin for a map of a mouse. Since we literally did not actually make a map, given that there are no spatial coordinates on this, it probably should be called a list of a mouse, but a list of a mouse in Latin sounds way worse, so we went with that. What is Tabula Muris? We have 7 different mice in the study, both male and female. Apparently, including female mice in studies like this is not a very common thing. We use 2 different methods to acquire the data. The first one I will be referring to as FACS. We didn't use FACS to acquire the data. We used FACS to sort cells in 34-well plates, and then we did SMART-seq2 full-length cDNA sequencing on those. This is a bit long, so I'll just refer to that as FACS. And then we used the microfluidic droplet method, available commercially by 10x Genomics, which I will be referring to as 10x or microfluidic, depending on my mood. Now, what this resulted in after sequencing and quality control is a list of approximately 100,000 cells, the numbers of cells per tissue. You can see in these graphs here. The top one is for FACS. The low one is for 10x. And these data come from 20 different tissues for FACS and 13 different tissues for 10x across the entire body of the mouse. So this is what basically our data set consists of. Now, beyond the reference, I would like to highlight at this point that the Tabula Muris is a multi-collaborative effort that was spearheaded by the Biohub, but it was not exclusively done by or even at the Biohub. And here you see a list of 16 different PIs. The labs whose helped a lot during the, both the analysis of the data, the annotation of the data I'm going to talk to you about, and also the actual experiments, sorting all the cells, and so on and so forth. And below each one of the pictures, you can see a little cartoon for the tissue that its lab was primarily responsible for. Now, these labs find themselves in UCSF and Stanford. There were also 4 different teams within BioHub that came together to do this. So my team, and then the teams of Andy May, Norman Neff, and Jim Carcagnas contributed to this work. The first thing we did, once we collected the cells, we made sure that their quality was adequate, was to try and figure out how we're going to annotate them. And by annotate them, I mean we need to give a name or a description to every cell so that for later analysis, we're sure we're comparing apples to apples and not to other fruits. Now, the cell annotation sometimes, from those of you that usually do this, is relatively complicated when you have to deal with one tissue. Now we have to deal with 20 tissues. This is something that, you know, it's beyond at least my personal biological expertise. So what we had to do is go back to the tissue experts and do this process that I'm going to describe to you with all of these 15 labs in basically real time, which can be also fairly complicated. So the annotation that I'm going to show you here and the way we did it was done separately for every method and for every tissue. This is important to keep in mind. I'll come back to that. What we do is pretty standard. We first start with the principal component analysis to project the most variable genes in the data set into a lower dimensional space. We cluster cells on this space, on this low dimensional PCA space. We produce visualizations using TSNE that, you know, can be used mostly for psychological reasons. We find differentially expressed genes for every cell cluster or every group of cells. These are basically, you know, genes that are expressed in this cluster more than they are in other clusters. And finally, we try to annotate each of the cell clusters using the list of genes using our existing knowledge of biology, and the annotation that we're using is either a free vocabulary, so you can call the cells whatever you like. You can call them, you know, John if you want, but given that this is not going to fly when we release the data, we also decided to use a controlled cell ontology vocabulary so that, you know, we all know what we're talking about. And if you do something similar, when we combine the data, you know, we know what cell is what, and we don't have to figure out that, you know, I call them B cells with a dash and you call them B cells with a dot, right? This is an actual example. So the annotation results in what you see here on the right. These are all the different muscle tissues that we have with FACS. Every little group of dots is basically a cell cluster colored by its cluster and obviously its annotation. So now we know what kind of cells reside in each of these tissues that we have data for. Now, if we are to build atlases, and if we are to move beyond them, and if we are to combine data from different atlases, this is really not going to work because we cannot analyze every tissue separately every time we probe a tissue. So the real question is, what happens if we take all of these and just throw them on the same graph, right? And what happens is this. Now, what you can see here is a TSNE visualization of all the cells in the data set, and they are colored by the tissue they come from, and the clusters are annotated by the predominant cell type. And what you can already start seeing is that there are 2 types of clusters. There are clusters of cells. Do I have a, whoop, I do. There are clusters of cells like the microglia that come from one part of the brain, which is good, or oligodendrocytes. And there are other cells that seem to be of the same type, so 2 different experts saw the cells in 2 different tissues and annotate them as endothelial, and when we put them all together, it doesn't really matter which tissue they come from. If they're endothelial cells, they tend to cluster with their neighbors, right, which are other endothelial cells. Now, this is great because it means that at this level of heterogeneity, the biological signal of what defines an endothelial cell is stronger than any technical signal that would separate this TSNE based on the mouse or the day or the person that did the experiment or the sequencer or whatnot. Now, having annotated the cells and making sure that another way to visualize basically what I'm showing you here is I can show you the same plot, and now it's annotated by cell cluster. And what we can ask is, what types of cells do we, oh, yeah, you have to take my word. What types of cells find themselves in which clusters? So what we're looking for here is one cluster, which could be this one, and then a group of 5, 6, 7 different types of T cells that come from different tissues, and they all find themselves basically being members of that cluster, which means basically what I told you before, that T cells will group together no matter where they come from. The same goes for endothelial cells, B cells, and a few other categories that you or I can't read right now. Having annotated the cells and knowing what we're looking at, we decided to have a quick look on how the different methods compare. I would like to say at this point that the main purpose of this paper was not to compare methods. If it was, we would have done a better job using methods that have comparable data. The 10x Genomics is using UMIs. The SmartSig method in plates that we're using is not. So an actual comparison is pretty tricky, but we can at least ask how many genes are detected in the same tissue, in the same cell type, using different methods. And what you see here, broken down per tissue, is basically the number of genes detected per cell, shown by box plots. Droplets, 10x is in red, FACS is in green, and what you see is expected and unexpected at the same time. The differences, the number of genes detected varies a lot between the methods, and the difference between the methods also varies a lot, with FACS coming out as the more sensitive method in most of the cases, but in many tissues, the droplet-based method doesn't have a problem matching at least the number of detected genes. In blue, you can also see an additional data set that was made available to us during revision from Han et al. With the microwell-seq data. Now, all right, so more genes are good, right? Well, it really depends on the question, and you cannot really answer without a question in mind. Obviously, for your money, yes, I guess detecting more genes is usually okay. So when it comes to describing the average cell population, and we can come back to that, I'm not claiming that the whole purpose of this exercise is describing average cell populations, but I'm just saying if that's what you're trying to do, and many people actually are doing that, what matters really is the total number of transcripts you can detect over the total number of cells you can analyze. Now, remember I told you FACS is more sensitive, detects fewer cells because it's more laborious, the 10x is kind of on the other side. So what we can do is that we can calculate average bulk profiles for all the single cell populations and then correlate them between the two methods. And what we see is that the correlation between the two methods with very different characteristics is generally pretty good. So when it comes to knowing what a B cell is doing, then both methods will probably get you there. Another way to view this is by asking how many of the differentially expressed genes in every cell do I pick up with every method? And again, the difference here is pretty big, but there always seems to be a yellow here, core of cell type specifying genes that is picked up by every method. So at the end, when it comes to information content for that particular exercise, we think that more or less the three methods we looked at are fairly comparable. Now, switching gears a little bit, what defines cell types? I've talked about that a lot. Remember I told you that our cell clusters were annotated using controlled cell ontology. Cell ontology has an inherent hierarchical structure built into it, so you can basically show all the relationships between all the cell types in our data set in the form of a tree. And then you can ask, what set of genes helps me recreate this tree, which for now I will consider to be kind of the ground truth, right? And what I'm showing you here is an example where we reconstruct this tree using the expression of only around 1,000 genes that are annotated as transcription factors. And this is how the two trees compare. And the entanglement of these two graphs, which should be 0 if the graphs are identical, is 0.1, which means that the transcription factors are doing a fairly decent job in reconstructing what we think we know about the relationships of these different cell types. Now, we can ask the same question using additional gene sets. So, for example, transporters, RNA splicing, cell surface markers, and whatnot. And when we do this exercise, we find that the set of genes that we use to annotate the transcription factors does the best job in restructuring the known cell ontology. So transcription factors appear important, right? They do. The question then becomes, which ones? So to answer this, what we did is the following over all the cell types in the data. We select one cell type every time. We compute a classification model of that cell type versus everybody else using random forests, using an algorithm that tries to basically find the smallest number of genes for a given classification error. We recover this set of transcription factors for every cell type, and then we try to compare that with what people know about these cell types. And what we find is, sorry, I'll need to run it again. There. What we find is that for cell types that I'm showing you here, like hepatocytes, skeletal muscles, this is the list of transcription factors that has the best performance when it's used to classify this cell versus every other cell. And what I'm showing you in color is basically genes that have been already reported in the literature by the references at the bottom of these graphs as candidates to be used for actual direct reprogramming of these cells from some sort of iPS or other embryonic cell type. So what we're hoping to do, or to get out of this, is within BioHub or for everybody else that has access to this data, try and see if we can refine or invent new reprogramming protocols using this resource. Now, another way of viewing that is you can ask what transcription factors help you differentiate between the same cell type across different tissues. So what are the differences between epithelial cells or endothelial cells that come from different compartments of the body when it comes to the transcription factors that help define them. All right, now I talked to you about our first commitment at BioHub and focus, which is understanding disease. I told you a little bit of what we're trying to do to get there, not just ourselves, but with others along the way. Our second commitment, as Steve mentioned in the morning, is accelerating scientific discovery. And to do that in the context of this project, we made sure that all the manuscript versions from the first one to the accepted one are on bioRxiv from day one. All the raw data are on GEO, and they've been there from day one, and they can be downloaded by anyone. All the process data in the form of gene counts and metadata are on Figshare, and the entire code that was used to collect, annotate, quality control, cluster, subcluster, anything, generate pretty much every figure in the paper can be found on GitHub. Now, easy access to the data for everyone is also important. Here I'm showing you a little demo of our web portal. And what you can do in this web portal is, without basically, need to program or write any line of code, you can check the expression of your favorite gene in the tissue you're interested with the method you're interested, and see your data in either a color TSNE or a box plot, violin plot, or download them as an actual table. All right. That would be the end of my presentation. I would like to thank Steve Quake, Tony Viscore, and their labs for the support and help throughout this project, specifically Nick and Fabio for helping during the entire course. I would like to thank members of my team for actually going in the lab and generating this data, which is not trivial, specifically Ashley. I would also like to thank members of Andy May's lab and Norma's lab, specifically René, for all the help. And finally, Jim Carcañas and his stellar team of data scientists that made pretty much most of the stuff that I showed you today possible. I would also like to thank everybody in the Tabula Muris consortium. And I would like to encourage you to visit our posters 6, 23, and 35 by Olga, Lincoln, and Angela later today. Thank you.
Okay, thank you, Spyros. We have time for a few questions. Yeah, please.
So I'm not exactly sure how you did the classification compute for the transcription factors, but very often it's really the combination of transcription factors that's decisive for a specific decision. So is this what you have done, or have you done it one by one?
No. So this is basically the entire set of transcription factors, and the algorithm tries to find the minimum set, right? So more than 1, 2, or as many as required for the smallest classification. So what I'm showing you is combinations. All of these transcription factors need to be used to be able to classify that cell from everybody else.
So in truth, when I remember the graphs for the individual cell types, what I have to imagine is that it's actually somehow the group of transcription factors, but they are somehow singly scored, so that.
So in the random forest classifier that I showed you, then we're looking at combinations. In the second graph that I showed you, where we're zooming in the epithelial and endothelial cells,
okay.
We're asking.
How many of the cell types can you explain by a specific combination of transcription factors?
I believe we find combinations of transcription factors for every cell type. It's just that the classification for certain cell types is not, perfect.
Right. Okay. Thank you.
Yeah.
Beautiful talk and project. I have 2 related questions, one related to Nikolaus's. So the first one is, what did you learn about power and biases? Meaning, how close is this to where we should be for any of these tissues versus how much more needs to be done? Mm-hmm. And whether you're losing some cell types or not recovering them and need to work harder for them. And the second, for the transcription factors, how important is the broader context to get the right combination? So if, for example, we dropped half of the organs and had to make do just with one half, would you have still gotten the same rough transcription factor code, or you only get it when you consider a really broad set?
Right. So the 2 questions are kind of related. I mean, you need to remember that most of the comparisons we're doing are someone versus something else. So it depends on what something else you have in your data. Right. Now, regarding the first question, so what we've learned so far, at least, is that with technology like 10x, which have all the downsides that I talked about, you can really get a representative view of what's happening on the tissue. You can capture rare populations. You can probably say something about fractions of cells. So if you're trying to get an overall look of, tell me everything that is there, and then, we'll dig in and figure out more details, then, being able to analyze large collections of cells is ideal. Most of the tissues in the FACS data set were actually FACS sorted to select cell populations that we knew already. So we didn't go in blind and just sorted everything, right? So, yeah.
So do you have a rough idea of where you would want to go in terms of scale compared to the 100,000 in the initial one, when you will start feeling comfortable for some of these tissues that you've recovered at certain rarity? Or
yeah, I mean, obviously this is kind of a circular argument, but I think for some of these tissues, we probably have a decent idea of what is there. So we're not going to find necessarily new subgroups of cells, not because they don't exist necessarily, but also the technology is limited, right, to some point. Right. Regarding the actual numbers and where do we see this going, again, I think for the mouse, probably increasing the numbers by a factor of 10 is not going to, revolutionize any of this, but maybe adding other perturbations, development, or, certain genetic models help you understand a little bit better. So I would probably move towards these directions next.
Cool. Thank you so much.
Thank you.
Okay, we have one last quick question, please.
All right. Amazing speak. I was wondering more of if within a certain cell population, can you distinguish like differentiation steps in that population, and then if that, it's different stages of differentiation of that cell, if that could correlate better in your, the way you correlate one cell in one tissue with the other tissue. So how can you address that question?
Given the age of these mice and given the number of cells we collected, we did not specifically look for like gradients of differentiation within these cell types. So to answer your question shortly, we didn't look. For some cell types, for example, in the brain, where you have the whole dance of OPCs and oligodendrocytes and whatnot, maybe it would make sense to actually start looking at these maps, but we haven't done that yet.